Hello, my name is Jefferson Smith, and I'm a final year PhD student at the University of Oxford. This is my poster titled Light-Activated Gene Expression in Synthetic Cells. So firstly, what do I mean by a synthetic cell? And as many of you will be familiar, I'm referring to compartments, in my case, a giant unilamellar vesicle, in which I've encapsulated a cell-free protein synthesis system and a light-activated DNA template. And so the idea is that in response to UV light, you get transcription of the DNA template into mRNA and then translation into the protein. And then the protein will govern the function of that compartment. So the light-activated DNA is as shown here, where the T7 promoter sequence, thymines shown in X, actually have this structure. They have a photocleavable biatylated linker attached to them, of which is then bound by monovalent streptavidin. And so a photocaged T7 promoter is created that represses the binding of a T7 polymerase. Upon irradiation with UV light, we deprotect the promoter, and this restores transcription and translation of the gene of interest. If we bring this together in compartments, we have light-activated amnion green DNA that won't express in the absence of UV light, but will then express in the presence of UV light. And if we take this a step further and pattern the UV light, we can actually spatially control the activation of synthetic cells according to the light that's used. So here, using a foil photomask, and here using a film photomask, we can pattern the synthetic cell populations. And we can also do more complex things. So, for example, we demonstrated communication between synthetic cells and E. coli through the in situ synthesis and release of an isovalerial homoserine lactone. So by expressing BJAI, the acyl homoserine lactone synthase, we can perform this reaction and convert S-adenosyl methionine and isovalerial-CoA, which are membrane impermeable precursors, into a membrane permeable isovalerial homoserine lactone. So this quorum sensing molecule can then activate gene expression inside E. coli through the expression of this BJAR protein, which regulates the expression of GFP from a PLOX or modified PLOX promoter. And so when we look at this data, we can see that the system works well. So in the absence of UV light, there is no BJAI expression, and so there is no lactone production. However, in the presence of UV light, the synthetic cells that are immobilized inside the agros will produce the isovalerial homoserine lactone, and this can activate gene expression in E. coli, as shown in these figures here. And so overall, the light-activated DNA can offer spatiotemporal control over gene expression inside synthetic cells. And looking further forward, we envisioned that light-activated synthetic cells could offer a platform for the controllable in situ synthesis and delivery of therapeutically active small molecules and proteins.